All right, engineering students. Okay, let's go to the second part of this video series on amino acids and proteins. So, want to proceed from where we left in the previous video, and what we want to say is that this this lecture on this one, and probably the one on the fuels, and also one on polymers will be the only three, no, including even the one on soaps and detergents, will be the only one in which we are going to be tackling aspects of reactions. But in general, in this course on chemistry 2, it will the course is entirely limited on nomenclature, the properties of organic compounds and their uses. But in this small part of the course, you need to understand some things about reactions, yeah, and nothing much. We're not going to talk about a lot about mechanisms, but in this case, what I want to say is that you can see here there is one amino acid that I put on the left and another which I put on the right. As I mentioned, for the amino acid, this is how you put it. The NH2 is on the left, the carboxylic acid is on the right. So even in the second one, I'm just going to put it the same way. The NH2 is on the left, and then the carboxylic acid is on the right. So how these amino acids they react, you simply put a box here and just say, hey, the first amino acid has got an OH which is going to be lost, and this one, the NH2 has got a hydrogen. So OH plus H are going to be lost as water. So these things are going to be removed. Yeah, you're only going to remove OH on one amino acids and the hydrogen on the left. Yeah. So the molecules which are going to the molecule you're going to remove will be a molecule of water. Now see here. So this bond and this bond, they're going to be joined together to form a what? To form a bond which are going to be called the peptide bond. Yeah. Can you see here? The rest of the things are going to remain the same. Yeah. Can you see this N, this H, hydrogen, Cr, this carbonyl group will remain the same. This other part, NHC, hydrogen R2, all the other part will remain the same. Nothing else will just change except the fact that that you're not going to be forming something called a peptide bond with the loss of water. So this kind of reaction is called a condensation reaction. Condensation means you are now precipitating water. Yeah? You're going to remove water. Yeah. So the final thing, so what you are forming from the two amino acids called amino acid residue. A residue is one which is formed from the initial compounds, and then you are removing one of the components, which in this case is water. Now, so in this case, what I've shown is that there's this first amino acid and the second amino acid that have been joined together to form a peptide bond. Now I've just shown here there was the only thing here which is changing is R. There is R1, R2, and then R3. You can get a case whereby R1 is the same as R2 is the same as R3. But what I've just done for the sake of this illustration is to say, what if R1 is different from R2 and is different from R3? So one amino acid and the next one, they are coming together to form this dipeptide. You can see here, nothing else is changing except the loss of water and then the joining of the one which I've shown in red. Yeah, are we clear on that? Now, with the other amino acids, now this avoids ambiguity. So if I've got the second amino acid, what I'm saying is that this amino acid is going to be joined on this other component, this other side as opposed to this other side. Yeah. Yeah. So in this case, if it will be joined here, this R3 is going to be next to the R2 because it will be joined on this side here. So the one which I've shown in red is showing it is the bond, it is the peptide bond which I'm showing in red here. It is the peptide bond which is shown as a result of the loss of water. Carbonyl group is losing an OH, and then the hydrogen, no, the amino group is losing hydrogen. So you're losing water. Also, in forming the second peptide group, you're also losing a molecule of water. So these are dipeptide. This one peptide bond, this the second peptide bond. So this one is called a dipeptide. Can you see that one? So can you see it's getting a bit complicated, but you can see there is R1, there is R2, and then there is R3. According to the structures of the amino acids which I shown you, is that when the R was hydrogen, we're calling that amino acid glycine. Yeah? You remember? And then when it was CH3, that amino acid, we're giving it the name of alanine. You remember? And then if it was SH, you are calling it serine. So this is what I'm just giving here. When R is hydrogen, we are calling it glycine. When R is CH3, we are calling it alanine. When R is SH, we are calling it serine. You're seeing that? So this structure which I've shown here, can you see here that the only thing which is changing is here. There is the hydrogen here, there is the CH3 here, there is the SH. Can you see that? So you can see from here, you can now show here. So this is glycine is joined to alanine, and then it's joined to serine here. Now, scientists, they like shorthand, and they don't like showing full name. So in your notes, you're going to see the three-hand shorthand for all the 20 amino acids. So glycine is called by the common name of GLY. Alanine is called ALA, and then the serine is called SER. Yeah. So in this case, so this dipeptide, whereby the glycine is joined to alanine is joined to serine, is going to be represented by the three-letter notation of GLY-ALA-SER. Yeah. Can you see it contains GLY glycine joined to alanine joined to serine. And then I put here some words here, very, very important. Even if it has the 3 amino acids, the order matters. Like in this case, what I'm saying is this: glycine joined to alanine joined to serine is not the same as say, hey, I'm going to put serine joined to alanine joined to glycine. You're talking about a totally different structure, which is what I've put here. In this case, I've shown here down. Can you see here that the first amino acid has got R is SH, which is serine. The next one is CO3, which is now alanine. The third one is GLY, is hydrogen, so it is glycine. So this amino acid is serine joined to alanine joined to glycine. Can you see the structure is different here? In this case, from the left, the structure contains hydrogen. In this case, from the left, it contains sulfur with hydrogen. And on the right, as you're adding here, this one contains sulfur with the hydrogen, and this one contains hydrogen. So the order of amino acid matters. The order of amino acids matters. Yeah. So the point which I'm trying to drive at is that even if you've got the 3 amino acids are present, the order in which they are joined matters. So glycine joined to alanine joined to serine is not the same as serine joined to alanine joined to glycine, even if they are the 3 amino acids present. Are you okay on that? Okay. So I think after a short while I'll go to the next part of this video series, of which I'll just show that that as much as we usually have this, we are showing it in this notation. When the amino acids are probably shown, if there will be questions dealing with amino acids, you're likely to be given the structures of the amino acids in an appendix. But you're not going to be given in this long notation, you're going to be given something called bond line structure. Bond line structure, like in this case, NH2, that will be shown here. Where there is the carbon and the hydrogen, it should be shown as zigzag. You remember those zigzag carbons. That's what we want to do next, just to show some of the amino acids. How can you show them in the bond line structure? This is the complete Lewis acid, but anyway, if it's the Lewis acid, you need to know, like the nitrogen has got a lone pair, sulfur has got 2 lone pairs, oxygen has got 2 lone pairs, nitrogen has got a lone pair here, this oxygen has got 2 lone pairs, nitrogen has got a lone pair here, oxygen lone pair, oxygen, you see like that. That is now the complete Lewis acid, has got those lone pairs, they need to be shown, yeah? But we want to show the bond line structure for the amino acids, and then we'll go to the to some of the properties of the amino acid. But once again, all these things are in your notes. This video series is not meant to substitute the notes, but just to clarify some of the things that needed to be clarified regarding the notes. So I'll just stop there, and then I think we'll move to the next part. Okay.